Let me grab my headphones, you guys. Welcome to the stream. A little bit of Bach. Welcome everyone. We'll start in just a minute. That was amazing. Any requests? I can only play free songs. Oh, Karma Kit. Hello. We're just giving people a minute to get here. We're going to be discussing how novel coronavirus affects your pets. Welcome to the stream, everyone. I can read your questions, by the way, if you have questions for me. I can answer questions. Hey, Krista. Just giving people a minute to get here. How is the sound, by the way? Can you guys hear me? Hey, Cameo. We'll start in just a minute, you guys. I know. Hey, Liam. How are you doing, sir? Miss you in class. Hey, Hamid. Man, yeah. I do miss having you guys in class and traveling to school and seeing you guys. The world has definitely changed. All right. Well, let's go ahead and get started. Looks like we have a few people here. Yeah, welcome you guys. Welcome everyone. So I found a paper yesterday was published in the journal Science, which is a very, very high level prestigious journal. And it has to do with pets, like domesticated pets. And the pets they researched in here were ferrets, cats, dogs, pigs, ducks, and chicken, basically. And they wanted to explore which ones of these domesticated pets can get infected with novel coronavirus. So it was a very interesting read, and they made some interesting observations, interesting findings, right? So we're going to talk about it. Why? Because we all love our pets, right? you guys, if you're familiar with my channel at all, you know about my cat Tig. We all love Tig, and he's getting older, so he's a senior cat. And, I'm concerned about him because I don't want him to catch coronavirus, just like I don't want seniors in our community getting coronavirus. Hey, Shadad, what's up, man? So, can our pets get sick? And that's pretty much the topic of today's presentation. Can our pets catch the coronavirus? Okay. And like I said, Tig is getting to be a senior cat, and I definitely don't want him getting sick because, we love Tig. So let's get started with this paper, right? Let's get started and see what they found. So this is the paper, this here. This is a paper published in the journal Science, and I told you guys that this journal is one of the premier journals in the world, right? So when you talk about top journals in the world, you're talking about the journal Science, you're talking about the journal Nature, right? And I think journal Science was actually started by Edison, if I remember correctly. So it's a very prestigious journal. And just yesterday, they released this research article. It's by Shi et al. It's Chinese researchers who were looking at coronavirus, and it's titled Susceptibility of Ferrets, Cats, Dogs, and Other Domesticated Animals. What they mean are pigs, ducks, and chicken, okay, to SARS coronavirus 2. What that is... Hey, Shelby, how are you doing? What SARS coronavirus 2 means, it's basically that's COVID-19. That's the novel coronavirus. By the way, before I move on, I just want to tell you the difference between, I just want to tell you the terms and what the terms mean, okay? So let me make this clear real quick. Let me go to scene 2. Hello. I think I have a notepad around here. Where did my notepad go? Oh yeah, here it is. I've got a notepad here, and I'm going to get me a dry erase marker because I want to show you something. Okay. Hello. So I just want to make something clear. All right. I don't think I even need these headphones. All right, so real quick, I need you guys to know that COVID-19, okay, COVID-19 is actually not the name of the virus. COVID-19 is the name of the disease, okay? What that means, COVID-19, what it means, it's coronavirus disease 2019. Okay, so when you hear COVID-19, they're not talking about the virus. Okay, they're talking about the disease caused by the virus. Okay, the virus itself is called SARS coronavirus 2. Okay, I didn't have enough room to write it, but SARS coronavirus 2 is the virus. COVID-19 is the name of the disease, okay? Just like HIV is the name of the virus, AIDS is the name of the disease, right? So you got to keep those straight. Now, sometimes we'll call things COVID-19 just because it's easier and people know what you're talking about. But just to be more accurate, you want to keep those terms straight. Anyway, so SARS coronavirus 2 is the name of the virus. And in this study, they were looking at how do these domesticated pets, these domesticated animals, come down with the virus or not. Okay, so first of all, we have to start with the abstract. The abstract is this big summary of what they achieved what they found in the story. So basically, in one paragraph, you're going to learn everything about their study. So this paragraph by itself is going to tell you everything they figured out. And then we can go into more details later, if that makes any sense, okay? So look at this. So, abstract. Severe acute respiratory syndrome coronavirus 2, that's what SARS-CoV-2 stands for. So the virus, coronavirus, the new one, causes the infectious disease COVID-19. Remember, so that's the disease. Okay, it's good so far. Which was first reported in Wuhan, China, December 2019. Despite the tremendous efforts to control the disease, COVID-19 has now spread to over 100 countries and caused a global pandemic. SARS-CoV-2, the virus, is thought to have originated in bats, because now we know that it has about a 96% similarity to bat coronavirus. Okay. However, the intermediate animal sources of the virus are completely unknown. So they don't know how the virus jumped to people, right? They don't know how the virus hopped to person. Like, what animal did it come from? They think maybe from a bat, then maybe to a pangolin, which kind of looks like an armadillo cat. Have you guys seen pangolin? And then maybe to a human. So that's what they're trying to figure out here. So here we investigated the susceptibility. That means if these animals can come down with the virus or not. Investigated the susceptibility of ferrets and animals in close contact with humans to the virus, SARS-CoV-2. We found that the virus, SARS-CoV-2, replicates poorly in dogs, okay, poorly in chickens, poorly in pigs, poorly in ducks. However, in ferrets and cats, it was permissive to infection. That means ferrets and cats can get infected, okay? Ferrets and cats can get infected, but dogs, pigs, chickens, and ducks, not so much, okay? That's kind of what they figured out. We found experimentally that cats are susceptible to airborne infection. Did you see that? So they figured out that cats can actually catch it just by breathing it in. But ferrets, not so much. Our study provides important insights into animal models for SARS-CoV-2, the virus, and animal management for COVID-19 control. Yeah, because think about it. If all you're doing is isolating people and you don't care what the animals are doing, well, the animals could be spreading it amongst themselves and the animals could be spreading it to the people. So you need to understand not just how to quarantine people, but also what animals is the virus hopping to, if you think about it, right? So this is their introduction. What is an introduction? An introduction is like a background. It's a general background to the field. Oh, you guys can't even see. Hold on. Let me switch back to scene 2. Basically, I just read this to you guys. Like I said, the virus can infect dogs, cannot infect dogs, pigs, chickens, or ducks, right? But it can infect ferrets and cats. That's what we were saying, right? So that's what they saw. The overall idea is that it can infect ferrets and cats, but it cannot infect dogs, pigs, chickens, or ducks. So let's get started. Introduction. What's the introduction? The introduction is an overview of the background, basically the background to the field. Okay, so what's known to this point, right? So let's get started. In late December 2019, an unusual phenomena emerged in humans in Wuhan, China, and rapidly spread internationally, raising global public health concerns. The causative pathogen was identified as a novel coronavirus that was named SARS-CoV-2 on the basis of a phylogenetic analysis of related coronaviruses by Coronavirus Study Group of the International Committee of Viral Taxonomy. The disease it causes was subsequently designated COVID-19 by the World Health Organization. Despite tremendous efforts to control COVID-19 outbreak, the disease is still spreading. As of March 11th, so this was actually written quite a while ago, as of March 11th, SARS-CoV-2 infections have been reported in more than 100 countries and 118,000 human cases. Now, it's obviously it's much more than that. This was back in March 11th. So I just pulled up for you the cases in the U.S. as of today. So this is CDC's website. As of today, it looks like we have, just in this country, almost 500 million cases and almost 15,000 deaths. So quite a lot has changed, obviously, in about a month, right? Because today's the 9th, and this paper was written, I'm sorry, let me switch this around. This paper was written, it was written closer to March 11th. So in about just under a month, you can see how much has changed, right? It's really ballooned because it's exponential. Each person infects an average of 2 other people. So that's what an exponential growth means. 1 becomes 2, 2 becomes 4, etc., right? So COVID-19 has now been announced as a pandemic by the World Health Organization. So although the virus, SARS-CoV, shares 96.2% identity with the at the nucleotide level with coronavirus RATG13, which is found in horseshoe bats. Did you guys see that? They found out that this novel coronavirus shares 96.2% identity with a horseshoe bat coronavirus, with the coronavirus that infects the horseshoe bat. So if I were to speculate personally, in my opinion, I would say that the coronavirus, that bat coronavirus, may have mutated to become this novel coronavirus. So somehow that bat virus may have mutated and now it can infect us humans. But that would be my guess, right? So in Yunnan province in 2013, it has not previously been detected in humans or other animals. The emerging situation raises many urgent questions. Could the widely disseminated virus transmit to other animal species, which then become reservoirs? Reservoir means an animal that harbors a virus. That's a reservoir. For infection, the SARS-CoV-2 infection has a wide clinical spectrum in humans, from mild infection to death, right? So people can get anywhere from mild infections to death, as a cause of this virus, right? So, but how does the virus behave in other animals? We don't know how this virus affects cats or dogs, right? As efforts are made for vaccine and antiviral drug development, which animals can be used, right? This is something scientists want to know. Which animals can be used to study the virus, right? Okay, and sorry, I have to make myself dance around so I can show you what I'm reading here. Most precisely to model the efficacy of such control measures in humans. To address these questions, we evaluated the susceptibility of different model laboratory animals as well as companion and domestic animals to the virus. All experiments were done with biosafety level 4, animal biosafety level 4 at this research institute in China. And were detailed, blah, 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 the protocols. I just wanted to show you, before I move on, I just wanted to show you how they did these studies real quick. Look at this. They weren't messing around. They gowned up completely in these hazmat suits, right? These biohazard level 4 suits, which are negative pressure suits. They are not risking it at all. You can see they've got their own air They've got no way of having viral contamination into their suits here, and they were working in this lab setting with these animals to protect themselves, and they were working in a negative pressure isolator room, which means it's like a vacuumed room. They would put animals in these cages here on the left, and then the air would be blown from left to right. So the air would blow, and then other animals would get exposed to those animals. So this is how they would infect animals. They would have animals that have the virus on this side, and then other animals on this side, and see if these animals would pick up the virus because they're downstream. The air would travel and infect these animals on this side of the room. So pretty interesting how they set up this experiment. All right, so let's talk about the experiment a little bit. So they started with ferrets, okay? They started with ferrets. Why did they start with ferrets? Well, some people keep ferrets as a pet, but the more important reason, the reason they really wanted to start with ferrets was that ferrets are commonly used as an animal model for respiratory viruses and how they infect humans. So when scientists study respiratory diseases, they often use ferrets as a model organism. Right. So this was a good way to start. We therefore tested the susceptibility of the virus in ferrets. 2 viruses, one from the environment. This was called F13E. They found coronavirus from the environment in the seafood market, and then they also found the virus in a human. Okay. So E means virus they found from the environment at the seafood market. H means virus they found from a human who was in Wuhan, right, who had the disease. So they got 2 sources of the coronavirus, one from the environment, one from a human, and then they used those viruses to infect ferrets, okay. Pairs of ferrets were inoculated. Inoculated means they were given the virus. Okay. Pairs of ferrets were inoculated intranasally. So that means like in the nose. So the virus was placed in their nose at this concentration, 10 to the 5 plaque-forming units. That just means how many viruses, a good number of viruses were stuck up their nose, of the environmental and the human virus. Then the ferrets were euthanized. Euthanized means put down, right? Like killed, okay? Euthanized on day 4 post-inoculation. So after they were given the virus. PI means after inoculation, after they were given the virus. Because we know 4 days after you're given the virus, you start to get sick, right? The virus is already replicating. Then the nasal turbinate. You know what the nasal turbinate means? It means the folds of the nose, the inside of the nose, you've got folds. That's the nasal turbinate. The soft palate, which means inside of your mouth, at the back of the top of your mouth where it's soft. That's your soft palate. Tonsils, your tonsils. Trachea, lungs, heart, liver, spleen, kidneys, pancreas, small intestine, and brain from each ferret were collected for viral RNA quantification by qPCR for virus and virus titration in Vero E6 cells. So what they wanted to do, here's what they did, right? They infected these ferrets with virus. Then, after 4 days, they put them down, and then they looked at how much virus is found in these parts of the body, right? The nose, the soft tissue of the mouth, the heart, the liver, the spleen, and also how much RNA is found in those as well. You know why they're looking for RNA? Because the genome of the virus is an RNA genome. This virus does not have a DNA genome. It's an RNA virus. So they're looking for the RNA and they're looking for the virus itself in those tissues. Okay, so let's check it out. So viral RNA, figure 1A and B. This is figure 1A. This is figure 1B. Okay, so in figure 1A, figure 1B, the infectious virus were detected in the nasal turbinate. This is nasal turbinate. Do you see that? You see how high these bars are? You see how high these bars are? The red one's ferret 1, the blue one's ferret 2, right? Look at that. You've got a lot of the virus RNA in the nasal turbinate, also in the soft palate, right? The back of the mouth, the top of the back of the mouth. You've got a lot of RNA. The tonsils have a lot of RNA. But then look at all the other tissues, the trachea, the lungs, the heart, the liver, the spleen, the kidney, the pancreas, the small intestine, the brain. Not much, nothing. You see that? So you know what they were able to conclude? This is what they were able to conclude, that the virus was able to replicate in the upper part of the respiratory system. What does that mean? That means in the nasal turbinate, the soft palate, and tonsils only. So basically, ferrets can only get infected up here. That's it. The ferret was not able to be infected in the lungs or anything like that. Interesting. And so this was looking at RNA, right? The RNA, the genome of the virus. And here they were looking at the actual virus. Like, could they find actual virus in those tissues as well? And you can see what they saw. The nasal turbinate, yes, it had actual virus. The soft palate had actual virus, and the tonsils had actual virus as well. You see that? The environmental virus and the human virus. You see that? But look at the other organs. Remember the heart, the liver, the pancreas, the intestines, all those other organs? There's no virus being made. So what this told the researchers is that only, and it tells you right here. It tells you right here. Here, let me move me so you can see it. These results indicate that the virus can replicate in the upper respiratory tract, meaning the nose and upper and the throat area of ferrets. But its replication in other organs is undetectable. So the ferret is only going to get sick here, right? That's it. Not in the lungs, nothing like that. So it doesn't really get all that sick. Okay, so that's kind of how it affected the ferrets. Okay, and then they went on and they did some more studies here. Let's just show you here some more studies. So let's take a look to investigate replication dynamics of these viruses in ferrets. So this was I'm not going to bore you with all this, but this is replication dynamics. What does that mean? They were looking at how quickly does the virus's RNA appear, right? So look, they took ferrets and they did it and they looked at 2 days after infection, 4 days after infection, 6 days after infection, 8 days, and 10 days after infection. And you see the virus RNA, it's already there at 2 days after infection. So this virus is quite quick. It gets in there and starts copying itself right away. You see that? That's what this. And by day 10, it has fought it off because, at day 10, the virus is pretty much gone. So these ferrets overcome the infection quite quickly. Isn't that interesting? All right, and look at this. This is viral loads. Whoops. Let me fix this for you. Yeah, this is viral loads. So this is the virus itself. And again, you see the virus itself peaks at around day 4 to day 6, and then it goes back down. So the virus peaks at day 4, and then a virus goes back down. So by day 10, the virus has pretty much disappeared. it's untraceable in the ferrets. Okay. And let me know if you guys have any questions. Is everything making sense so far? Are you guys understanding this stuff? Let me know if you have questions, because I know it can get kind of convoluted, and I'm trying to make it as straightforward as possible. A lot of it is, pretty confusing if you've not seen this kind of stuff before, but I'm trying to break it down as much as possible. Let me know if you're confused at all. All right, so let's keep going a little bit. Another thing they wanted to look at was the infectious virus was detected from the nasal washes of all ferrets. So basically, they used saline, washed out the noses of the ferrets, and they were able to find virus in the noses of the ferrets, especially at day 4. You see, at day 4, you can find lots of virus in the ferret nose. But when they look at rectal swabs, so basically they wanted to see if the RNA is present in the poop, right, of the ferrets. They weren't able to find any RNA in the rectal swabs from the ferrets. So the virus is not going through the digestive tract. Now, one ferret from each virus inoculated group developed fever and loss of appetite on days 10 and 12 post-infection. That's pretty interesting. Let me move me again so you can see. So, whoops. So look here. One ferret from each group of 3 became loss of appetite and developed fever. Okay, to investigate whether these symptoms were caused by viral replication in the lower respiratory tract, they euthanized the ferrets at day 13 and collected their organs. However, the viral RNA was not detected in any of the other tissues. So again, they were able to show that, look, on day 13, you only see the virus in the nose. You don't see the virus in any of these other tissues. So those ferrets are getting fever not because they have severe respiratory issues or pneumonia, but probably just because they're fighting off this infection from the nose. So then they went on and they looked at the lungs of ferrets, and they were able to see some kind of damage to these lungs and some kind of macrophages and neutrophils. These are white blood cells coming to the lungs. So there was some damage to the lung, but they don't understand how this damage was happening to the lungs, especially since the virus itself wasn't infecting the lungs, right? So that was kind of strange. Now, on day 13 post-infection, okay, antibodies against SARS-CoV-2 were detected in all of the ferrets by an ELISA and neutralization assay. So you don't need to know what ELISA and neutralization assay are, but what you need to understand is they found antibodies against the coronavirus in all of the ferrets, which means that the ferrets were fighting off the infection. They got the infection and then they were fighting off the infection. Whenever you're fighting off an infection, you form antibodies, okay? And these researchers found the antibodies using these techniques, ELISA and neutralization assay. That indicates that these ferrets fought off the infection. That's what that means. Although the antibody titers of the 2 ferrets that were euthanized on day 13 PI were notably lower than those of the ferrets euthanized on day 20. And that makes sense. By day 13, your body has mounted less of an immune response than at day 20. At day 20, you're going to have a lot more antibodies than at day 13. You see this? Look here. So these are 3 different ferrets, right? 3 different ferrets, red, blue, and mustard, okay? These 2 ferrets were euthanized at day 20, so they had a lot of antibodies in their system. This This ferret was euthanized at day 13. So it has far less antibodies in its system. So they're just saying that antibodies were found in all of these ferrets, but the one that was sacrificed earlier at day 13, not at day 20, it had less antibodies, which makes sense because it takes about 2 weeks to start ramping up your antibodies. And by day 20, obviously, you're going to have more antibodies against the virus than at day 14, right? So that makes sense. Okay. They also looked at the lung of the ferrets, and they were able to see some kind of a little bit of scarring in the lungs, but mainly when they looked for viral RNA, which is what they care about, the viral RNA in the tissues and organs, viral RNA was only found in the nasal, turbinate, and soft palate. So the viral RNA was only in the nose of the ferrets and in the soft palate, the top of the back of the mouth. So interesting stuff. Okay. And then what else? So here they're looking at these results indicate that SARS-CoV-2 can replicate in the upper respiratory tract of the ferrets for up to 8 days without causing severe disease or death. So basically, to wrap up their studies, they showed that these ferrets do not get severe disease. They do not die from the infection, but they can get infected. Look at this. They do get an infection of the upper respiratory tract. What does that mean? The nose, the top of the mouth, you know, the inside of the mouth, the tonsils, the trachea, these parts, right? And after day 8, you don't see the infection anymore. So up till day 8, you see the infection. By day 14, the infection's gone. So these ferrets can get infected, but they cure themselves by day 14. It actually looked like they cured themselves by day 10, remember from the previous figures. And they quickly build antibodies and they fight off the infection. They don't really get all that sick, and they don't really get, they don't die, right? Or at least not the ones they studied. They probably should have studied more. Ideally, they should have looked at, I don't know, like 50 ferrets or something, but they only studied some. I don't know exactly how many ferrets they studied, but maybe we could figure that out at the end. So that's ferrets. Okay. So now they're moving on to cats. This is probably what we all care about more, especially what I care about more because, you know, I care about Tig, my cat, and, you know, I care that he's okay. So, you know, this was the part I was really interested in, was dogs and cats, and that's next. Cats and then followed by dogs. So let's talk about this. Cats and dogs, Cats and dogs are in close contact with humans, and therefore it is important to understand their susceptibility to the virus and to the disease if we were going to control the disease. We first investigated the replication of SARS-CoV-2 in cats. 7 subadult cats. So subadult, what does subadult mean? That means cats aged 6 to 9 months old, outbred domestic cats, were intranasally inoculated. So they were given the virus in the nose at this concentration, 10 to the 5 plaque-forming units, PFU, of the human-derived virus, 2 animals were scheduled to be euthanized. That means put down on day 3 post-infection and day 6 post-infection, respectively, to evaluate viral replication in their organs. So then they were going to figure out, is the virus growing in their organs? Is the virus dividing in the organs? 3 subadult cats were placed in separate cages within an insulator to monitor respiratory droplet transmission. An uninfected cat was placed in a cage adjacent to each of the infected cats. Did you see that? So look at this. They put the cats that were given the virus in these cages here. You see these 3 cages? Cage, cage, cage. They put cats that were infected in these cages. And then they had a fan. I read this earlier. They had a fan, and the fan was blowing air. You see the arrows? That's air movement throughout this room. You see what I'm saying? So the air is moving this way through the room. And then they had other animals, uninfected animals here. So they had like healthy cat here, healthy cat here, healthy cat here. You see what I'm saying? So, again, infected cats here, healthy cats here, and then the air was blowing, and that's supposed to indicate transmission, right? Transmission. Okay. Droplet transmission. Remember I told you what droplets are? When you cough or sneeze or even talk, you can release these mist particles. Those are droplets, and those droplets harbor the virus, right? An uninfected cat was placed in the cage, blah, blah, blah. It was difficult to perform regular nasal wash collection on the subadult cats because they were aggressive. Oh, jeez. So, yeah, I guess if you jammed a bunch of virus in my nose and put me in a cage, I'd probably be aggressive too. To avoid possible injury, we only collected feces from these cats and checked for viral RNA in their organs after euthanasia, after they were put down. So they were quite afraid of these cats. They were overwhelmed with fear, and they were overpowered by these cats. By the way, if you have any questions, just let me know. If you're getting confused at all, or if I go too fast on something, just let me know. I'm reading the comments, so, let me know if you have any questions at all. But anyway, interesting, right? So let's see what happens with the cats. So here's some cat data, all right? Viral RNA, remember, that means the genome of the virus. Viral RNA was detected in the nose, the nasal turbinate, that means the folds of the nose, of the inside of the nose of one animal, in soft palates, tonsils, trachea, lungs, and small intestine of both animals that were euthanized on day 3 post-infection. Look at figure 2. Look at figure 2. Day 3 and day 6, look what you have. The virus is in the nose, the soft palate, the tonsils, the trachea, and the small intestine of these cats. And look at this. By day 3, it's in the lungs as well of these cats. It's in the lungs of the cats as well. So in the cats, unlike the ferrets, remember in the ferrets, you were only able to infect the ferrets in the nose, the palate, the tonsils, and a little bit of the trachea, right? So the upper respiratory tract, right? But in cats, it looks like they were infected in all of that, as well as the lungs itself and the small intestines. The small intestines. That's kind of weird. Okay. So let's go back to this. In the animals that were euthanized on day 6, basically it was the same, except there was no indication of viral RNA in the lungs. See, so at day 3 you have viral RNA in the lungs, but by day 6 you don't have viral RNA in the lungs. It's almost like you fought it off in that short amount of time. Infectious virus was detected in the viral RNA positive nasal turbinates. Oh, okay, so these graphs here on the right, this is not just viral RNA, it's infectious virus. It's the virus itself. So look here, the virus itself was also found in all of these tissues, at day 3. So in day 3, you found virus in the nose, the soft palate, the tonsils, trachea, and the lungs. But by day 6, it looks like it's cured from the lungs, but it's still present in the upper respiratory tract. So it looks like cats, they can actually fight it off from their lungs faster than they fight it off from their upper respiratory tract. What else do we know about cats? So in the transmission study, viral RNA was detected in the feces. So they monitored the feces, right? They monitored the poop of the cats. So what did they see in the transmission study? So this means, remember they had a healthy cat downstream of the infected cat. Viral DNA was detected in the feces of 2 virus-inoculated subadult cats. And in all 3 virus-inoculated subadult cats at day 5, that makes sense because these were the cats that were given the virus. You see, the cats that were given the virus have the virus. And then the cats that were next door that were healthy, one of them got the virus. You see what happened, you guys? You see this? You see this one bar here? This means that a healthy cat picked up the virus. This guy. The rest of the healthy cats did not pick up the virus. But this healthy cat picked up the virus. You see that? And you know where he picked it up? He picked it up in its nose, in its palate, in its tonsil, and trachea. Okay, interesting stuff. So you know what that means? You know what these results tell you? These results tell you that cats can spread the disease. Cats can spread it through just breathing or coughing or whatever they're doing, right? So a cat can pick up the infection, a cat can get infected, and then a cat can, through, droplets, infect another cat. Okay, I don't know. They didn't show if it can infect a person, but it can affect another cat. Isn't that interesting? So now they were looking for antibodies. Remember what antibodies are suggestive of? If you find antibodies, that means that the cat is fighting the infection. Does that make sense? So they found antibodies for the virus in all 3 of the inoculated cats. So look, you see these bars? They found antibodies against the virus in these cats. These were the cats that were given the virus. And do you know what this cat is? This is the cat that got the virus from the other cats just by those cats breathing on this cat. You see, these cats breathed on this cat. This cat got the disease, and he's fighting the disease off too. So he's building antibodies. So this poor cat picked up the disease from just being next door to these cats, and this cat is fighting off the disease. You see that? He's fighting off the disease. So what does that tell you? Cats can pick up the coronavirus. Cats can get infected by the coronavirus, and cats can infect other cats. And then once you're infected, you fight off the disease. And when you fight off the disease, you have the antibodies against that. So we replicated the replication and transmission studies in juvenile cats. Okay, and so they did it in juvenile cats, so the younger cats, and they saw the same stuff. You infect the cats, and look, they get infected. The viral RNA is in all of these tissues: the nose, the throat, the tonsils, trachea, lungs, right? And by day 6, it's getting a little bit abated, a little bit lower. We replicated the transmission studies. Oh, this is with the viral RNA. They did it in juvenile cats. And again, look, these are the cats that you gave the virus to. You see, these were the cats that you gave the virus to. You see how these cats have the viral RNA, and then these cats exposed these cats, and then you see this cat here. This cat got the transmission as well. So not just older cats, but also juvenile cats can transmit the virus through breath, through respiratory droplets. Okay, so we replicated the transmission study in juvenile cats, and look at this. viral RNA in organs of juvenile cat that died on day 13 post-inoculation. So one cat died, okay? One cat died. So they wanted to figure out why. So they found a lot of the viral RNA in the trachea, the breathing tube, right? The trachea is the tube that goes down to your lungs. Okay, the trachea had a high concentration of viral RNA in the cat. And then they wanted to figure out what was wrong with this cat. Why did this cat die? So they did histology on the cat. And this is what they saw. They saw in the tissues of the cat, they saw problems, right? So this is figure A. Figure A is nasal respiratory mucosa, abnormal arrangement of the epithelium, and loss of cilia. I don't know if you guys watched my previous video on this paper. Remember, this was the novel paper from China, the first paper showing that this disease is occurring, and this is the paper that showed that coronavirus is a novel virus. But do you remember in this paper, one of their findings was that, you see here, the virus causes problems in the cilia, the little hairs that beat in the lung cells, right? The little cilia of your lung cells get damaged by this virus. So if you remember that, this won't be very surprising. What they found in the cats is that there's loss of cilia in the lung cells, the lung epithelial cells. That means the lung skin cells of essentially of the cat. This little cilia, these little beating hairs are getting damaged, right? And you need those little beating hairs in your lungs to help clear your lungs of debris. Okay, what else? The tonsil epithelia had degeneration and necrosis. That means there was some damage and death in the tonsils. In the trachea, there was degeneration and necrosis. More degeneration and necrosis in the trachea. The lungs had inflammation. More inflammation in the lungs. Okay, lung infiltration of macrophages. So here, macrophages are your white blood cells, and these white blood cells were infiltrating the lungs. So this is indicative of what happens during a severe lung infection. This is what happens. The white blood cells really try to get in there and eat the virus, and that can cause problems like pneumonia. So I wouldn't be surprised if this cat had pneumonia. There was a lung intra-alveolar edema. So the alveola are the tips of the lungs, the little sacs that fill up with air where the blood meets the air, and there was actually bleeding there. That's not good. And then lastly, or second last, the small intestine had degeneration. Remember, the small intestine was infected by the virus as well. The nasal turbulate, you see, oh, look at where it's brown. Do you guys see where it's brown? This is a picture of, they sectioned the nose of the cat, right? They sectioned the nose of the cat. And wherever you see brown, that's where the virus is replicating. Okay, that's where the virus is. So you can actually see the virus in the cat nose cells, in the turbulates, right? Turbinates, I should call them. You can see the viral antigen, so the virus itself in the nose. In the tonsils as well, you see in the tonsils, you see the virus growing, and you see some degeneration of the cells. And in the small intestine, you can see the virus in brown. The virus is growing and degeneration of those cells as well. All right. So now, moving on to dogs. Some of you have dogs and or care about your dogs. We all care about our dogs. So what's up with dogs, right? Dogs. Can dogs get coronavirus? This was interesting to find out. We know cats can, like poor Tig. So they next investigated the replication and transmission of SARS in dogs, right? 5 3-month-old beagles were intranasally inoculated with 10 to the 5 PFU of the human virus. So they put this amount, again, the same amount they've been putting in the ferrets and the cats. They put this amount of virus in the nose of the beagles. And the version of the virus they put was the human version, the one they got from a human. And then they also put it with 2 other beagles, 2 uninoculated beagles. Yeah, like Odie, right? So again, what did they do? They stuck the virus in the nose of one beagle, and then they housed that beagle with 2 healthy beagles, 2 beagles that they did not infect, okay, in a room. Oropharyngeal, that means back of the throat, and rectal swabs from each beagle were collected at days 2, 4, 6, 8, 10, 12, 14 after infection for viral RNA detection and virus titration in these cells, Vero E6 cells. Viral RNA was detected in the rectal swabs of 2 virus-infected dogs on day 2 and in the rectal swab of 1 dog on day 6. So look at this. So they treated the dogs, and the inoculated dogs, you see 2 of them had rectal swab RNA. 1 of them had rectal swab RNA on day 6. 2 of them had day 2. 1 dog that was viral RNA positive by its rectal swab on day 2 was euthanized. So they killed him on day 4. But viral RNA was not detected in any organs or tissues collected from this dog. So look at this. Even though they were able to get the RNA from the rectal swab, so, a rectal swab from the dog, when they looked at the dog's organs, its nose, its palate, tonsils, trachea, lungs, heart, liver, spleen, kidney, pancreas, small intestine, brain, they couldn't find any sign of the virus, not the RNA of the virus, not the virus itself, right? Or here, actually, they were looking at just the RNA of the virus. They couldn't find the RNA of the virus. Also, infectious virus was not detected in any of these dogs either. So look here. They looked for the actual virus itself, and they couldn't find it in any of the dogs either. Sera were collected from the dogs on day 14 for antibody detection by use of ELISA. 2 virus-inoculated dogs had antibody. Okay, so 2 of them had antibody. Look at this. 2 of them made antibody. Remember what antibody is indicative of? If a dog is producing antibody, that means that its body recognized the virus and it fought it off, right? So 2 of the dogs made the antibody against the virus. The rest of the dogs didn't. But for whatever reason, the dogs didn't show any of the actual virus inside, right? They didn't have the viral RNA. Very strange stuff. So what did they conclude? These results indicate that dogs have low susceptibility to the virus, to the novel coronavirus. Okay, so if you own a dog, you can be more relaxed than if you own a cat or a ferret, right? Cats can get the disease and spread the disease to other cats through respiratory droplets. But dogs, looks like they don't really come down with the virus. They're not a good reservoir for the virus, which means the virus doesn't really grow all that well or replicate. Susceptibility of pigs, chickens, and ducks to coronavirus using the same strategy as that used in to assess dogs. However, viral RNA was not detected in any swabs collected from these virus-inoculated animals or from naive contact animals. So basically, they weren't able to find any sign of the virus in any pigs, chickens, or ducks. So pigs, chickens, and ducks pretty much immune to the virus. So now off to the summary. We're almost done. Thank you for bearing with me and learning something. In summary, what did they find? In summary, we found that ferrets and cats are highly susceptible to the coronavirus, the novel coronavirus. Dogs have very low susceptibility, and livestock, including pigs, chickens, and ducks, are not susceptible to the virus. Ferrets have frequently been used as animal model for study of human respiratory virus. Unlike influenza virus and other human SARS coronavirus, which replicate in both the upper and lower respiratory tract of ferrets, we found that the novel coronavirus only replicates in the upper respiratory tract of the ferret. Remember, not in the whole respiratory tract of the ferret. So ferrets aren't going to be the best model organism to study if you want to study coronavirus, but at least they kind of get infected by the virus. It may also replicate in the digestive tract, as viral RNA was detected in the rectal swabs of the ferrets. But virus was not detected in the lungs, even after ferrets were intratracheally inoculated with the virus. So even though they were inoculated directly into the trachea cannot get infected by the virus. So the somehow only the upper respiratory system of the ferrets gets infected by the virus, not the lower respiratory tract. It remains unclear whether the virus causes more severe disease in male ferrets than female ferrets, as has been observed in humans. In humans, the coronavirus seems to cause more damage in males than in females. Several studies have reported that SARS-CoV-2 uses angiotensin converting enzyme 2, so ACE2, as its receptor to enter the cells. ACE2 is mainly expressed in type 2 pneumocytes and serous epithelial cells of the tracheobronchial submucosal glands in the ferrets. Ferrets and cats have only 2 amino acid differences in the SARS-CoV-2 spike-containing region of ACE2. So what does all that mean? If you guys watched my second video or third video on this topic, you would have seen that the way that coronavirus infects your cells is that coronavirus has these proteins on its surface called spike proteins, and those spike proteins attach to your cells, and they specifically attach to the cells that have this protein on them called ACE2. If your cells have this ACE2 protein on them, coronavirus will attach to that cell and infect that cell. So what they're saying is cats, the only difference between cats ACE2 and ferret ACE2 is just 2 amino acids. Look, here and here. So ferrets have a tyrosine and what is this, like a glutamine or something? What is R? Tyrosine? No, glutamine, tyrosine, threonine. I think R means threonine. Ferrets have threonine and tyrosine here. So maybe because their ACE2 is slightly different than our ACE2, maybe that's why ferrets are immune to getting infected by the coronavirus. But, you know, it says it still has to be investigated. They don't know for sure. It still needs to be investigated there. Okay, so the respiratory tract of ferrets remains to be investigated. The fact that SARS-CoV-2 replicates efficiently in the upper respiratory tract of the ferrets makes them a good animal model for evaluating antiviral drugs. You could study antiviral drugs, but you have to be aware that you're only going to be studying the upper respiratory system of these ferrets, not the entire respiratory system. The cats they used in the study were outbred and were susceptible to the virus, which replicated efficiently and transmitted to naive cats. So if anything, cats are a better model organism to study if you want to study coronavirus, because cats can get infected not just in their upper respiratory cells. Arginine. Okay, yeah, thanks, Shadad. So R is arginine. So this cat has, ferrets have arginine here and tyrosine here, and that's the difference they have in their ACE2 receptor. that might be why their cells are not as susceptible as ours, because our cells have a histidine here and a glycine here, right? So what that means is our ACE2 receptor looks a little bit different than a ferret's ACE2 receptor. You see what I'm saying? And that little difference might be the difference between the virus being able to attach to it or not, if that makes any sense. But anyway, cats have been reported to be seropositive with the virus. So the cats can have the antibodies for the virus. And so what they figured out from this study is cats can catch the virus. Cats can get sick in their upper and lower respiratory tract and their intestines. And cats can spread it to other cats. So you need to surveil for the virus in cats, not just in humans. You need to be also factoring in cat-to-cat transmission and possibly maybe cat-to-human transmission as well. So that about sums up the paper. I hope you learned something. Again, let's just quickly summarize. What does it mean for you if you have pets, right? If you have pets, just be careful, because apparently cats can come down with the virus, though it doesn't seem to be as bad in most cats. I definitely would want to keep Tig away from the virus. I wouldn't want him to get sick. Okay. It's definitely something to look out for if you have a cat. If you have a dog, it's not so big a deal. Looks like they're pretty resistant against the virus, pretty immune to the virus. So, it does give me cause for concern, with Tig, especially since Tig is a senior cat and we love him dearly as a member of the family. So I will be, watching out to not spread it to him. That makes me wonder, how deadly is the virus to cats? They didn't study many cats here in this study. And they definitely didn't study cats as old as Tig. I think the oldest cat they studied in this paper was 9, right? Whereas Tig is like 16, So, what about cats Tig's age? How susceptible are those cats? Are those cats even more susceptible? It might be something in the future that when the vaccine comes out, you may have to vaccinate your cat, in the future against coronavirus. Otherwise, the cat might, become very sick and die, or it can become sick and transmit it to other cats. Who knows? I could see a future where you not only get, when you get the shots for your cats, perhaps you'll get a coronavirus shot for your cat as well, especially since they could transmit and become sick, and no one wants their pet to get sick. So very interesting stuff. Again, cats can get sick. They can transmit it to other cats. Ferrets can get sick, but they, get less sick. They only get in the upper respiratory tract, but other pets seem to be fine. So hopefully you learned something interesting. That was a really interesting novel paper. Thank you guys for joining me. It's always fun. And I'll try to do something like this again pretty soon for you guys. Send me, if you guys find any papers like this, any interesting papers you want me to review and discuss, just let me know. Maybe send it to me, shoot me a message, and I'll be glad to do that for you guys. Thank you so much for joining me, and I'll catch you guys next time.